There we go. Okay, if somebody would lower, please, thank you. Okay. So this is where we were last time. This is where we ended. Docking vessel, synaptic tagmin, smear proteins. Everybody understand that concept? What's coming down here? How this, the calcium is just not magic. Right? The calcium causes a conformational change in the vesicle, and because of these two proteins, right? The calcium actually binds here and then causes this docking protein to be activated. Right? Okay, so this goes through a lot of what we talked about last time. Remember that this is exocytosis that's occurring here. So you can read this. Okay, this is something interesting. So this is removal of neurotransmitters. So going back here, oops. Let's pretend, again, we're just using the neuromuscular junction as an example. Okay? This could be happening between two neurons, neuron and glial cell, neuron and endocrine cell, but we're going to just use the neuromuscular junction. So if this is the neuromuscular junction, remember that this neurotransmitter is acetylcholine, right? So ultimately, the acetylcholine will be exocytosed into the cleft here, and it will bind to the receptors, right? And the receptor for this particular neurotransmitter is an ion channel, right? And it's going to let in sodium, so that's why you see this channel here. But the process does not last forever, right? It has to be reversed somehow. And part of that reversal process is the removal of neurotransmitter. There's a whole long reversal, okay, but this is part of it. To terminate the signal in a chemical synapse, the neurotransmitter must be removed. Okay, it must be removed. If you don't remove the neurotransmitter, and there are actually drugs that can potentiate this. And when we get to muscle, we're going to talk about some diseases where this actually occurs, and you have to use a drug that allows the neurotransmitter to stay attached to the receptor longer. Okay? So you can have diffusion of the transmitter from the cleft. So some of it's just going to move away. Okay, it's just going to diffuse away. Some of it's not ever going to get to the receptors. Degradation. So there are enzymes that physically take the molecule apart while it is attached to the receptor. There are enzymes that physically break it to pieces. Once it's broken into pieces, the receptor is then deactivated. And then you have to, even when you break it up, you have to reuptake. So this is the body recycling. So if you can imagine that this is acetylcholine here. After the muscle contraction has occurred and it no longer has to occur, an enzyme is going to come in here. It's going to break this into actually 2 or 3 different molecules, and then it's going to diffuse out. There are actually processes that occur here, back on the presynaptic terminal, that will reabsorb the parts. So endocytosis will occur. Phagocytosis, right? Penocytosis, remember those? And they will be taken back up, those parts. What's going to happen is those parts are then going to get transported back towards the soma by what? What system? What is that?
Dynein and kinesin.
Dynein and kinesin. Right? Up that microtubule pathway, and then those parts are going to get put back together, then they're going to get put back into vesicles, and it's going to get shot back down again. And so your body does this over and over and over again. Dynein up, kinesin down. Yep, that's correct. So you're constantly recycling, right? You can make the stuff de novo, meaning without this, but why would you do that? Right? When you can just recycle it, just keep reusing it over and over and over. Okay. Activation of an excitatory postsynaptic cell. So when we're talking about a postsynaptic cell, right, it could be a muscle, a gland, a neuron, lots of different things. Mostly we're talking about a neuron and a muscle. You can have what are called EPSPs and IPSPs. These EPSPs and IPSPs have to do with what are called graded potentials. Remember the graded potentials? There's a list, a table with all the different kinds. So this stands for excitatory postsynaptic potential. So you should know that. And then this one stands for inhibitory postsynaptic potential. All right. So what does that mean? That means that when you have certain neurotransmitters, which we're going to look at, bind to a receptor, it doesn't mean that it will immediately cause an action potential to occur. Sometimes just the binding of that neurotransmitter to the receptor will cause a tweak up or down. Remember when we looked at that? The little tweak up and then the little tweak down? Now, some neurons are called excitatory neurons. Some neurons are inhibitory neurons. For instance, the motor neurons that go from your spinal cord or your brainstem to your muscles are only excitatory. So they cannot be inhibitory, meaning that whatever they release has to be excitatory. Now, can those motor neurons be influenced by inhibitory or excitatory neurons above them? Yes. Yes, they can. Okay. And that's where botulinum toxin and tetanus toxin come into play. Because one affects inhibitory neurons that bind the muscle motor neurons, and one affects excitatory. So the actual motor neuron itself that connects with the muscle is only excitatory. But what comes into it can be multiple things. So we're going to see some examples of this. So here is an excitatory postsynaptic potential, right? This is a slight depolarization, which means it goes from a negative 70 to, negative 65 here. It's becoming slightly more positive. Okay, and why does this thing tail off? Do you remember, what did he say? If this is a type of graded potential, why does this thing tail off? What did we say about graded potentials? Yeah, here you go.
As it gets further away from the source,
It degrades. That's why it's degraded. Can an action potential do that? Does it do that? It doesn't. Maybe if you were, 10,000 feet long, it would, but not in the human system. Okay. So this is an inhibitory, same thing. So this type of signal is coming from an inhibitory neurotransmitter. This type of signal is coming from an excitatory neurotransmitter, released from an excitatory neuron, where this one would be released from an inhibitory neuron.
which is what release the opposite. If the neurons release the opposite of the are.
No, the neurons will release the inhibitory neurotransmitter. So an inhibitory neuron will release inhibitory neurotransmitter, and an excitatory neuron will release excitatory neurotransmitter. Now, what they affect may be, an excitatory neuron may synapse with an inhibitory neuron and have an excitatory effect on that. But the effect isn't always what you think it is. Sometimes it's reversed. That's where it starts getting confusing. But in its simplest form, this is probably an inhibitory neurotransmitter. Okay? And the funny thing is that acetylcholine, right, which is an excitatory neurotransmitter at the neuromuscular junction, at other places within the nervous system can actually be inhibitory. So it gets a little confusing sometimes. And that has to do with the receptors that it binds to. Okay. So here's an example of a neuron that is being affected by other neurons. Okay? So this neuron is synapsing with maybe 3 neurons here. So here's an axon from 3 different neurons. In C, in red here, this synapse here is inhibitory. So whatever is being released here causes the tweak down, causes this to occur in this neuron. Whatever is being released in the greens is actually causing these upticks. So whatever the neuron is, probably within the central nervous system, it's the combination of how many excitatories there are and how many inhibitories. And they can balance each other out. And if you want something to activate fully, then you're going to have more excitatory input than inhibitory. Does that make sense? and they add. So if you only get one excitatory signal every whatever this time frame is here, then you're only going to keep getting the same depolarization. If it happens more frequently, right, then you start getting an increased depolarization. If it happens very, very frequently, you then get the threshold, and then you will have this go to firing an action potential. Does that make sense to everybody? So you can tweak the system, right? But once an action potential is activated, can you stop it? It is impossible. It is all or none. But until you get to that line, you could do all kinds of crazy things all day long. Up and up and up, up all day. Oh, not enough. So sorry. Come back down. But you can keep playing this game all day long. But as soon as you reach the threshold, you fire an action potential. It's all or none. It's either all on or all off. So it's on here, and this is what happens. You get a spike, and that's how you measure it. If you had an instrument sensitive enough to do this. Okay? That's what this is showing you. So EPSPs and IPSPs are a form of graded potential. So they have all of the properties of other graded potentials. All right. This is another example. This is an axo-axonic. So not only can you have a synapse between an axon and either a dendrite or the soma, but you can actually have a synapse an axon that synapses with another axon. Okay. So if I ask you what types of synapses can there be, there's lots, right? You can go synapse, axon to axon, axon to dendrite, axon to soma. All right. And so you can see that this would be the presynaptic cell, right? And this would be the postsynaptic cell in this synapse. But in this synapse, this would be the presynaptic cell, and this would be the postsynaptic cell. And their receptors are different colors because they respond to different neurotransmitters. That's what those different colors mean. That the molecule that's binding here is different than the molecule that's binding here, and is different than the molecule that's binding here. All right, so this is, again, for you, you can read through this. It has a lot of this it puts it in an outline form for you. So there's lots of things you should look at here. But we're not going to. All right. So does everybody understand that system? Very, very basic. Because now what we're going to do is say, if you have drugs or disease, how do you modify the system? Okay. So I'll read the very first one. It says drugs act by interfering or stimulating normal processes in the neuron involved in neurotransmitter synthesis. So the presynaptic cell, storage, again, the presynaptic cell, and release, presynaptic cell, or receptor activation. This could either be presynaptic or postsynaptic depending on whether it's an autoreceptor, right? Remember, the autoreceptor is the one that's on the cell that releases the neurotransmitter. Diseases. All right, so one of them is tetanus. We talked about this the other day. We talked about it again. Everybody's had a tetanus shot, right? You have to have a tetanus shot in order to come to college. Right? You have to have one every 10 years. It protects you against Clostridium tetani, and it produces tetanus toxin. Okay? Tetanus. So how do you get it? The bacteria lives in soil. It lives in dirt. Now, a lot of people have the misconception that you can only get tetanus from stepping on a rusty nail. It's not the rusty nail that's special. It's the fact that the bacteria can hide on the rusty nail. All kinds of nasty stuff can hide on a rusty nail, right? But there's nothing special about a nail. So if you're playing on a construction site and you get poked by a nail, and they go to the emergency room, the first thing the doctor or the nurse and NP or the PA is going to say, what happened? You say, well, I was playing on a construction site, I got poked by a nail. The first thing they're going to ask you then is, did you have a tetanus shot? If you haven't had a tetanus shot, they're going to give you one. If you've had a tetanus shot, they won't give you one. If you're not sure if you had a tetanus shot, they're going to give you one anyway. Because this is what they're trying to protect against. So what does it do? It prevents the vesicle fusion with the membrane. So when we go back to that diagram with those vesicles that are coming down the kinesin system, right? Those vesicles are floating around in the end of the axon. Normally, when they're activated by calcium, they're going to fuse with the end of the axon, right? That's what we said, that's what we showed. This toxin prevents this. It inhibits the neurotransmitter release and causes increased muscle contraction, right? So it prevents vesicle fusion. So this toxin affects inhibitory neurons. That's important. It affects inhibitory neurons because what comes next, if you don't understand that, makes absolutely no sense, right? It says that it prevents vesicle fusion with the membrane. Ultimately, it causes increased muscle contraction. And you're saying, well, if it inhibits a neurotransmitter from being released, how can it increase muscle contraction? Right? Because what it's doing, if you think of it this way, remember this is a muscle cell, like this, muscle cell, right here's the junction. This is an axon, it's myelinated, right? It's a motor neuron, right? What it's doing is it's happening here. So the tetanus toxin is being taken up here or here, and in this area here, it's preventing this negative. Can this neuron here be anything but excitatory? No, but can it be affected by excitatory and inhibitory neurons? Yes, and it is. So you have all kinds of neurons that are coming from your spinal cord and your brainstem fusing with motor neurons. And when you see a negative, that signifies that this synapse is inhibitory. If you block this, right, then that means that this thing is going to get closer to depolarization all the time. Now, you're not going to just do this with one, because you may have, 1, 2, 3, 4 inhibitory neurons that are being blocked. And they're controlling the firing pattern. And if you take away that control mechanism, then, every time if you're looking at this graph, right, and this is in millivoltage, let's say this is at negative 70, right, and let's say this is threshold. Every time you take one of them away, you do this. You take another one away, you do this. Take another one away, you do this. If you do it enough times, eventually you're going to get the threshold and you're going to fire. The problem is that even if you repolarize it, it's still going to go right back to threshold. And that's what tetanus does over and over and over again. So if you get tetanus, you're essentially paralyzed because you're over-contracting every muscle in your body. Eventually, it keeps getting worse and worse and worse. And like I said, you'll snap every bone in your body. You'll destroy your joints. You'll snap your vertebrae. Everything. You'll snap them all eventually. But that's not what's going to kill you. This is like the torture before you die. What's going to kill you is you're going to stop breathing. So that's why it happens, but it's happening here. Okay? Now, how is that different than botulinum toxin? Okay, Clostridium botulinum is the most potent toxin known. It's nasty. Nasty, nasty. Okay? It's Botox. That's what Botox is. Right? It interferes with the action of snare proteins at the excitatory synapses. Okay, so where would be that? If we're looking at a motor neuron here, this is an excitatory synapse. So those snare proteins, right? These snare proteins, right? This is synapto. This one is synaptotag. Synaptotagmin. And then this is the snare protein here. So it interferes with these snare proteins in excitatory neurons. So, at excitatory synapses that activate muscles, botulism is characterized by muscle paralysis. So they're essentially doing something very similar, but they're doing it to two different types of neurons. Tetanus toxin does it to an inhibitory neuron and causes you to be overactive. Botulinum toxin does it to the neuromuscular junction and causes paralysis. That's why it works, right? You take it, you inject it into your, if you have crow's feet as you age, in your head, and you inject it into the muscles at a very, very low concentration. This is why you don't want to have some hack do this. You don't want your hairdresser doing this. Not a good idea. What it does, it will paralyze those muscles, and it relaxes them. And that's how the wrinkles go away. You know if somebody's got Botox, because depending on where they've had it injected, it's paralyzed. It doesn't move anymore. It's pretty easy to tell. Once you know, you'd be like they laugh and they're moving their face and nothing crinkles up, they got Botox. Because it causes paralysis. Now, if you do this enough, in enough different places, and diffusion occurs, and if you get it into your blood, it can have, bigger consequences. So you don't want any hack doing this. And there's a certain interval that you can do it at. There's a certain concentration. It should be done by a medical professional. What is it, like $250, $500 a shot? Lasts like 3 months, something like that. So if you got the money, it's fantastic. Whatever you want to do. Be like a Kardashian. All right. Low doses of botulinum toxin, Botox are injected therapeutically to treat a number of conditions, including facial wrinkles, severe sweating, uncontrollable blinking, misalignment of eyes, and others. So there are other medical uses of this, right? But it's so diluted. It has to be because it's so dangerous. Whoever came up with this is brilliant. They thought this can kill people, but if I do it this way, I can make millions and millions of dollars. And they have. This is almost as good as Viagra. They made almost as much money as Viagra. I still think Viagra probably made more. Okay, questions. You get this? Does this make sense to you? So you can use a toxin from a bacteria to do good things or bad things. It's neither good nor bad. It depends on how you use it. All right, so drug effects. So this just goes through some of the effects. So there are all kinds of things that the drug might. So this is a good list of things. So we'll go through this. You can increase the leakage of the neurotransmitter from the vesicle, right? So more of it can leak. B, you can increase the neurotransmitter release in here, so you can have more of it coming out here. C, you can block the neurotransmitter release, right? So you can prevent it from opening up. That's one way to affect it. D, you can block, or you can inhibit the transmitter synthesis, which would be, they're showing it down here, but it would be back in the soma, right? So you can prevent the synthesis of this stuff from occurring. That's one way to block it. You can block the reuptake, meaning that the recycling process. You can bind the receptor to the postsynaptic membrane to block an antagonist, right? So you can have another drug that binds here. It's called competitive inhibition. That is the opposite of what this does. Or a drug that will bind here and do zero. So it's just enough, the same size and shape, that it binds this thing, but it doesn't actually do anything. It just sits in there. Or you can have another molecule that we've synthesized that can do the same thing as this drug. So let's say you don't make enough of this transmitter, you can inject some synthetic in there that will activate this. You can also inhibit or stimulate second messenger activity. So this is just what's happening here. You can also do stuff on this side, on the postsynaptic side, to affect whatever happens downstream. So there's lots of things you can do with drugs. This is what medicine does, right? To affect things in a good or a bad way. So the more you understand about the system, the more interesting the drugs become that you can create. But you have to understand the physiology first, right? You have to understand the molecular biology first, and then you use that information to help. Hopefully most of the drugs we make are helpful. Okay. You can go, then we're going to go through this list, but let's talk about a few different kinds. So these are classes of chemicals presumed to be neurotransmitters. Okay. So You have acetylcholines in its own class. Then you have what are called biogenic amines, catecholamines. You ever heard that in a hospital setting? The catecholamines are dopamine, norepinephrine, and epinephrine. That's the group of three that are called catecholamines. Serotonin, serotonin reuptake inhibitors, Prozac is a serotonin reuptake inhibitor. Right? So anything that people would take for bipolar syndrome or depression, those are usually serotonin reuptake inhibitors. Serotonin is the feel-good neurotransmitter. Right? If your serotonin is really low, anybody ever been on a low-carb diet? One of the reasons you feel so miserable, there's a lot of reasons why you feel miserable on a low-carb diet, but one of the reasons is you're not taking in carbohydrates, and you need carbohydrates in order to synthesize serotonin, and serotonin is the feel-good neurotransmitter, right? So if you don't have any, you're going to be more likely to be unhappy. That's why people that are dying, if you tell me you're on a low-carb diet and I have anything to do with you, and you're not eating carbohydrates, I don't want you anywhere near me, because you're going to be miserable. You are. Because you're not making serotonin. Serotonin makes you feel good. You're not going to be happy ever. Okay? Not a good idea. And then histamines. Histamines are also neurotransmitters. And they're called biogenic amines. So this is a class of them. You have another kinds of neurotransmitters that are just amino acids. They're not even full proteins. An example of this is glutamate. Glutamate is the main excitatory neurotransmitter in your body. Okay? Glutamate. And then there are inhibitory. GABA, gamma-aminobutyric acid, is one of the main inhibitory neurotransmitters in your body. So you can mess with these two neurotransmitters and have really profound effects on your physiology. And glycine is also inhibitory. You have neuropeptides like opioids like opium, heroin, morphine are all opioids. Okay, they're in that class, neuropeptides. Oxytocin. Oxytocin is a hormone/neurotransmitter. It is the hormone that causes contractions to occur for delivering. It is a bonding hormone. Okay, it's stored in the posterior pituitary, but is also a neurotransmitter. And tachykinins. So all of these are involved in, we think, the runner's high. If anybody's a runner here that's ever had it, there's some evidence that suggests that all three of these molecule types are involved in that, where you sort of feel this euphoria. Like, you can't be hurt, it's all great, life is good, you want to wax poetic, right? So these are all involved in that. Then you have gases. Nitric oxide, carbon monoxide, and hydrogen sulfide. In addition to everything else that they do, you can actually use them as a neurotransmitter. This neurotransmitter we're actually going to look at when we look at dilation of blood vessels. Highly involved in dilating your arteries and your veins. And it's a gas. And then ultimately you have purines, and purines, adenosine and ATP. The adenosine receptor is the one that caffeine works on. Caffeine actually is a competitive inhibitor of the adenosine receptor. The reason that you get jacked up with caffeine is because the adenosine receptor is inhibitory. When you have a lot of adenosine binding to the adenosine receptor, you're tired. If you block the adenosine receptor with the molecules that are in caffeine and coffee, you then become awake. So you're messing with this system, the purine system. So you should know that there are 1, 2, 3, 4, 5, 6 classes of neurotransmitters/neuromodulators. Okay, what is a neuromodulator? So I'll read you their definition, and I'll give you my definition. Neuromodulators modify both presynaptic and postsynaptic cells' response to specific neurotransmitters, amplifying or dampening the effectiveness. The neuromodulators, the way I define them, is they're like that switch there that has a dimmer on it. They tweak things one direction or another. A neurotransmitter usually turns something on or off. But a neuromodulator is more of a tweak type molecule. It's going to amplify a little bit or make it more difficult for that neurotransmitter to expand. And a lot of these molecules that we just talked about can be one or the other. There's a lot of research needed on this. We just don't know. Okay. Another thing that neuromodulators can do is they can affect metabolic processes. Okay, so some channels, some receptors that neurotransmitters bind to are called metabotropic. Metabotropic. And some are called ionotropic. Here, it's mostly ion channels that are being affected by ionotropic neurotransmitters. Here, it's mainly G proteins, but not exclusively. And these metabotropic usually are affecting the metabolism, whereas the ionotropic are not. So, they're just two different types. Questions on that? Again, a lot of this is here for you to read through it in your own time to understand it so that you can ask questions if you don't understand it. All right, so acetylcholine we've talked about a little bit, right? It's found in the peripheral nervous system and the central nervous system. Neurotransmitters, neurons that use the acetylcholine as their primary neurotransmitter are known as cholinergic neurons. So acetylcholine acts, there's two different subtypes of receptors that it acts on. There's a really good diagram in your book of this. Okay? The muscarinic receptors are G protein coupled receptors. Most of these are located within the central nervous system. Okay? If you look at the chapter, they talk about the central nervous system, the brain and the spinal cord, and then they talk about these ganglia, right? The petrosal ganglia. These ganglia were these groupings of neuron cell bodies outside of the central nervous system that are sort of this midway point between the central nervous system and other organ systems. Within that system, you have these muscarinic receptors. You also have nicotinic receptors. The nicotinic receptors you find at the neuromuscular junction. So this one you're finding outside of the central nervous system. The other place that you find muscarinic receptors are in, we haven't learned about this yet, but in the autonomic nervous system has the sympathetic and the parasympathetic nervous system, and the parasympathetic effects are due to muscarinic receptors. You find these in blood vessels too, so that blood vessels, when they activate a muscarinic receptor, those blood vessels will dilate. Okay. So this is how acetylcholine is produced. You have this enzyme called choline acetyltransferase, CAT for short. Acetyl-CoA, if you're taking biochemistry, right? It's part of glycolysis. Acetyl-CoA comes out of glycolysis, feeds into the Krebs cycle, right? And you can use acetyl-CoA to do lots of different things. So acetyl-CoA combines with choline molecule. Where do you get choline from? Food like eggs contain high levels of choline, right? And then you form acetylcholine plus CoA. That CoA can then do lots of different things. Okay? So this is how you make it from scratch. Take acetyl-CoA, it comes out of glycolysis. Some of it goes into Krebs, some of it goes and does other things. You combine it with choline, you make acetylcholine and CoA. You degrade it. Remember we talked about an enzyme degrading once it's bound to the receptor, the neurotransmitter. And so that enzyme is called acetylcholinesterase. It's a mouthful, or big A, big C, small h, big E. And what it does is it breaks acetylcholine that's bound to the receptor into acetate and choline. Now notice that. Notice that the two molecules it breaks it into are different than the molecules that build the thing. So these two molecules get reabsorbed by the neuron that produced it. The choline is ready to go, but this acetate has to then get converted into acetyl-CoA before it can recombine and form acetylcholine. So these are not exactly the same. There's more processing that has to occur once it gets reabsorbed back into the neuron. So once it gets reabsorbed back into this neuron, the acetate and the CoA get transported up. The acetate gets converted via the metabolic machinery of the cell back in acetyl-CoA, combines with choline, and then ships it back down here, and it's ready to go. Questions on that? Any of that? I like this section because we hear about all these toxins, these nasty things. So, have you ever heard of sarin gas? Ricin is another one. There's mustard gas, really, really nasty things. Sarin gas is considered a weapon of mass destruction, by the way. So is ricin, and so is mustard gas, right? That's why we went into Iraq, right? Many years ago after 9/11, because there was stuff like this, right? Sarin, nasty. So some chemical weapons, such as the nerve gas sarin, inhibit acetylcholinesterase, causing a buildup of acetylcholine in the synaptic cleft. So when it builds up in the cleft, you have two problems. One, it makes that muscle contraction continues to occur, and two, it can't resynthesize it, right? So it can't uptake it. So that junction here, this thing is always active. And that can't be, right? Because you can't repolarize the cell. Again, how does it kill you? Well, there's lots of nasty stuff that it does to you, but ultimately you can't breathe anymore. But it does all kinds of nasty stuff, so it's sarin gas. Overstimulation of the postsynaptic acetylcholine receptor causes uncontrolled muscle contractions. People start going into convulsions, ultimately leading to receptor desensitization and paralysis and death. That's how sarin gas would do. Any questions on that? So lots of natural. So sarin gas we produce, right? The tetanus toxin and the botulinum toxin are naturally produced by the organism. Sarin gas is something that we, in our infinite wisdom, have decided is a great product to make. So, fantastic, right? Best scientists in the world figured this out. All right. Nicotinic receptors in the brain are important for cognitive function and behavior. The presence of nicotinic receptors on presynaptic terminals in reward pathways of the brain explain why tobacco products are among the most highly addictive substances. The tobacco contains thousands of chemicals, right? But one of them is nicotine. And we know a lot about nicotine, and when you smoke, if you've ever done this, if you do this, then you get a hit in your brain that releases dopamine via this receptor system. Dopamine is the pleasure hormone. So every time you smoke, you get a hit. That's why people who smoke, when they get nervous, right, or really stressed out, they got to go have a smoke, right? It makes them feel better, and it's hard to give it up. And so that's one of the reasons why it's so hard to quit smoking. Because you get a feel-good experience every time you do it. And why would I do that? Plus, the other thing it does is it activates and elevates your metabolic rate. If you want to see what happens, try stopping smoking. Let's see how many pounds you put on. A lot of people, when they stop smoking, they'll gain 10, 20, 30, 40, 70 pounds because of this. Part of it is the slowdown in the metabolism, but part of it is they're trying to find that hit, the dopamine hit from something else. So you have to find something to replace that feeling, and it's not an easy feeling to come across. That's why it's so hard to. Questions on that? Sorry, yes, or Alzheimer's disease, another really interesting phenomenon. So neurons associated with acetylcholine system degenerate in people with Alzheimer's disease. So in Alzheimer's disease, it's a very complex disease. It all happens between the ages of 65 and 85. It causes these neurofibrillary tangles, tau proteins, prion-like substances. These things are non-functional. Okay? And what ends up happening is these people, this cholinergic system breaks down, so they can no longer, they start to not be able to feel pleasure anymore, right? And that makes people angry, your inability to do this. So because, and I'll just read, this one's a really good one, I think we should read it. So because of the degeneration of cholinergic neurons, this disease is associated with decreased amounts of acetylcholine in certain areas of the brain, even loss of postsynaptic neurons. These defects and those in other neurotransmitter systems that are affected in this disease are related to declining language, perceptual abilities, confusion, memory loss, and the ability to feel pleasure. So a lot of times when somebody's having an episode of Alzheimer's or dementia, they're related, they're not exactly the same, they become very ornery, right? And part of it is that they have less of an ability to feel pleasure anymore, so they become more and more angry. So if you know someone has Alzheimer's, they can become extremely violent, and sometimes they need to be sedated because of this kind of stuff that's going on. Plus, the higher level areas of the brain, the frontal cortex, which means you do, are also being affected, so they don't rationalize very easily either. They become very violent. Okay. This is just, we went through these before. The only thing I want you to focus on in this slide, so you have that table that we went through. The enzymes which degrade the biogenic amines are monoamine oxidase and catechol O methyltransferase. So if you have biogenic amines, okay, remember the biogenic amines are dopamine, norepinephrine, epinephrine, serotonin, and histamine. These can be degraded by one of these two. So this is the same type of molecule, an enzyme, as acetylcholinesterase is. Acetylcholinesterase breaks down acetylcholine, MAO, and this one here, the transferase, break down these molecules. If you've ever read any research regarding norepinephrine or dopamine or serotonin, oftentimes they talk about MAO because this is the enzyme that breaks it down, right? That enables it to break away from the receptor, be taken up by the cell that produced it, and recycle it. So oftentimes they use these in the experiments. So if you come across that, that's what that is, MAO. So you should know these. The list here that we've already gone through. You're not going to have to know the structures. Okay? You're not going to have to know the structures. But this is the pathway. You should know the pathway. So you need tyrosine, right? Tyrosine hydroxylase is the enzyme that converts tyrosine to L-DOPA. Dopa decarboxylase converts L-DOPA to dopamine, which is then converted by dopamine beta hydroxylase to norepinephrine, and then it's converted to epinephrine. So these catecholamines are all produced in the same pathway. This is the pleasure hormone, right? These two are excitatory neurotransmitters. They're part of the fight or flight response. They're all produced in the same pathway. So likely, if you have a problem producing these two, you probably have a problem producing this one. So in cocktails that they give people who are depressed, remember with serotonin reuptake? They have cocktails that they give people now. One of them is a serotonin reuptake inhibitor. One of them affects the reuptake of norepinephrine and epinephrine, and the other one, this is the brand new one that they're just starting to use, affects the reuptake of dopamine. Because you can't just affect one, they're all screwed up. They're all in disproportion. And that's why people who have these issues, you know, they're not only are they depressed, but they can't feel pleasure and they can't get excited. It's because these two and serotonin are all out of whack. So you can't just affect one. So this is the problem with physiology. In human beings, once you affect one molecule, you're probably going to affect a whole lot of others. This is why a lot of scientists don't like working in the physiological realm. I love it. They don't like it because it's complicated. Because in reality, physiology is complicated. They would much rather take a cell, take DNA, and mess with it in vitro, right? It's so much easier. You ask a question, you totally can control it. The minute you put something in a living system, a rat, a mouse, a hamster, a goat, a horse, a human, you have no idea what's going to happen because every change you make may have 10 additional changes that are down the line. So you never really know exactly what you're doing. So when we create drugs, why am I telling you this? So when we create drugs, when you go on medication, right, a lot of this has been studied. But even with stuff that's been studied, there's always side effects. Of course there are. Of course there are, because the minute you mess with one of them, you screw up a bunch of others. They don't know them all. It's super complicated. Very, very complicated. So it is a big deal, you know, when you're on any type of medication, because it's the side effects. It's the collateral damage that that drug causes. You would be better off on being zero most of the time. Now, that doesn't mean that if somebody's depressed, they shouldn't be on an antidepressant. That's not what I'm saying. But what I'm saying is that it would behoove you, if possible, to try to do additional things and try to get off it, if possible. If not, obviously that's between a person and a psychiatrist or a psychologist, but it would be better because there's always collateral damage. Does that make sense? Always going to be collateral damage. And this showing you, this is a simple version of it, right? If these two are screwed up, this one probably is too. Or if this one's screwed up, then maybe these two are screwed up. These are really important molecules that affect how you act and who you are. And if they're messed up, then you're not going to act the way you normally do. So it's a big deal. So you don't have to be able to write the chemical structure, but you should understand this pathway. You should know, you know, the different molecules and the enzymes that go into creating this. Questions on that? Don't write the molecule fraction. Right? I'm not going to use it. If you like to write and do that, that's fine, but I'm not going to get it. Okay. Adrenergic receptors are the receptors that respond to epinephrine and norepinephrine. This is really, really important because when we get to muscle and we get to cardio, we're going to keep referring back to these. All right, so we have to understand these receptors. Super important. Okay. Adrenergic receptors are G protein coupled receptors. You know what a G protein coupled receptor looks like, how it works, right? The different pieces. There are two classes. There's alpha and betas. There's alpha 1 and alpha 2. This is the Greek notation, right? And then there are beta 1, 2, and 3. When we get to muscle and when we get to the vessels, blood vessels and the cardiovascular system, we keep referring back to this over and over and over again. So this isn't the first or the last time you're going to see this. I mean, it's maybe the first, but it's not the last. Excuse me. So, generally speaking, these are excitatory and these are inhibitory, but not excitatory. When you think of a neurotransmitter and its effect, you got to start thinking more like a cell biologist or a molecular biologist. And start thinking the effect that it has is determined by what's downstream from the receptor. What do I mean by that? If there's a receptor here, right, that binds acetylcholine, right? What the effect that this receptor is going to have depends on what comes after it. You can have the exact same receptor and neurotransmitter, but if that receptor subtype is slightly different, So instead of being like that, it looks like that. Then what's downstream may be also different. So the same molecule doesn't have the same effect in every cell in the body, is what I'm trying to say. So you have to think about it as, okay, an alpha receptor, how many different kinds of alpha receptors are there? And if norepinephrine binds to a 1 versus a 2, are we going to have a different effect? Sure. Yeah. Are the receptors a little bit different? But is the neurotransmitter that bound them the same? Of course. Of course. So the same neurotransmitter will bind all 5 of these, and yet it has at least 5 different effects. So you should know all of these receptors, the names. We're going to come back to them. Parkinson's disease. Let's talk about another disease. So Parkinson's involves dopamine production. People who get Parkinson's disease don't produce enough dopamine anymore. So they start having a tremor. So you start seeing someone who's Parkinsonian, they have a Parkinsonian tremor. Right? Because it's an excitatory neurotransmitter. They have head nodding, so it looks like they do this a lot. They do tremors. Okay? Bent forward, shuffling gait, stiff facial expressions, slow initiating and executing, like Muhammad Ali has Parkinson's disease, right? They form of Parkinson's disease. A lot of famous people have it. The dopamine producing neurons in substantia nigra, which is one of the places that produces dopamine, they're shutting down for some reason. And they produce this excitatory neurotransmitter. And so when that excitatory neurotransmitter is not being produced in enough quantities, all of these different things can happen. So it is currently treated with the drug L-DOPA in the initial stages. Now, let's go back. L-DOPA. This is how they're boosting it. They make an artificial form of this, and you take it. And then this will upregulate the production of dopamine by giving this. It's not perfect, but it helps. Right? And that's because the more of this you have, this is the raw material, the more of the hormone you can make. But it's not perfect. It doesn't cure it. There is no cure for Parkinson's disease. So that's what L-DOPA is. Another thing that they're doing now for Parkinson's disease is called deep brain stimulation. They take these electrodes, they connect them to your skull, and then they have these frequencies that they push into your head. And for some reason, that seems to help these neurons. Another thing that can help prevent Parkinson's disease, coffee. It has been shown that drinking coffee up to 4 cups a day will decrease your chances of getting Parkinson's disease by 45%. We don't know why. 45%. So if you drink lots of coffee, you're 45% less likely to get Parkinson's disease. It's interesting. We have no idea why or how it works, but something about the caffeine or the coffee, it's probably not the caffeine, is good. Right? So there's an interesting topic, right? Serotonin. So serotonin, again, is also abbreviated 5-HT, same thing, because serotonin is 5-hydroxytryptamine. It is also made by these weird cells. You don't have to know this. I'm not going to have you. This is a mouthful, right? Enterochromaffin cells. And 90% of the serotonin is made in your GI system. The other 10% is made in your brain stem. It's made by serotonergic neurons in the medullary raphe. So drugs. These SSRIs we talked about are drugs that people are on, including Prozac is one of them, if you're depressed or bipolar syndrome. LSD stimulates a subtype of serotonin. So LSD is not a hot drug nowadays, but in the 70s, LSD was And the reason people took it is because they got the serotonin in it to make you feel good. Right? So a lot of people were on LSD. What does serotonin function in? Regulating sleep, emotions, breathing. In the field that Dr. Miller, Justin Miller, and I are from in neural control of breathing, serotonin we look at for CO2 sensitivity to understand how your body responds to carbon dioxide. It regulates growth and vascular smooth muscle. So serotonin is one of these neurotransmitters / hormones that's involved in thermogenesis, heat regulation, a lot of different things. And so it's not just about when you don't eat enough carbohydrates, about feeling good, but the other things it's involved in also start to go. You can't sleep very easily, right? If you're on a low-carb diet, your sleep sucks. I'll tell you right now. One of the best ways that they have shown to get a good night's sleep is if you don't want to eat carbs all day long, get practical. I'm all about practical. If you eat a big carb meal right before you go to sleep, sleep like a baby. One of the reasons is that you need the molecules in the carbohydrates to make the serotonin to help you sleep. Right? So if knowing a little bit of the science actually helps. But yeah, if you don't have enough serotonin, you're miserable. And you'll never be happy. So it's not a good thing. Histamine. Histamine, we talk a lot about it with allergies. Right? But it's also an excitatory neurotransmitter. A very type of old histamine blocker is Benadryl. Anybody ever take Benadryl here? You have allergic reaction, right? A little bit of a rash. Benadryl is a histamine blocker. What happens if you take Benadryl? Let's say you take the maximum dose of Benadryl. What happens to most people? They go to sleep. They go to sleep because it's blocking histamines, and histamines are excitatory neurotransmitters. Now, the new kinds of histamine blockers, like Claritin, don't do this. So they found a way around it. So Claritin is non-drowsy, but Benadryl will knock you on your butt because it's blocking histamine production, and the histamines are excitatory neurotransmitters to help keep you awake. Now, there's another type of histamine in your gut, and there are different receptor subtypes that helps produce the acid, the hydrochloric acid in your gut. Okay, I'm going to stop here. I just want to talk quick about the exam. We have one more lecture. We'll finish this up, and then we have the exam on Friday. So on the exam, a couple of things. You need to come in with strategy, right? You're only going to get an hour and 5 minutes. I will make sure that I am here early. So if you want to start a few minutes early, is there anybody in here before us? Yeah. Okay, so we'll try to get in here as early. Are they usually done early? It seems like it. So if you want to start a little bit earlier, I'll come. Unfortunately, I don't know if there's anybody after this class. There may not be, but my second class, I think, there is. And so what I do for one group, I have to do for the other. So that's one strategy. On the exam, as far as it's all lecture material up through nervous, no muscle. We didn't get to it. I'm not going to tell you to read it and do it on your own. Okay. So no muscle. But the Physio X portion of the exam will go through muscle, right? There are 3 labs that you had to do. Apparently, there's this misconception going around that it's optional to get Physio X and that it's not important. Unless I was talking Mandarin Chinese, you can look in the syllabus. It says required. Required means you have to have it, and you have to do them. They're not optional. So if that's a misconception, you now have 4 days to fix it. Get Physio X, do the labs, because they're going to show up on questions. I don't know why that is. I couldn't have been more clear, but I'm trying to save your butts right now. Good thing that somebody told me just about 5 minutes before I came in here. All right, you have to do Physio X. It's in the syllabus. It even says it on the exam. Physio X, labs 1, 2, and 3. So, okay. Questions? Yeah, here.
For some of the Physio-X, or actually all of them, they have those like short answer questions at the end. Do you know if there's like an answer key for any of those? Because sometimes you write them and you're not sure if you watched.
I have an answer key. So if you have questions on it, you should talk to me and Miller. Yeah, they have them. Yeah, that's a good question. Yep.
Going on with that, so when we do that, is that the type of questions that we should expect?
Yeah, so that's a good question. So it'll be questions like that and questions about, so there's two types of questions, right? One is like that, but here's the thing. I know you can go on the internet and find the answers to those because I've seen it. So it exists. So if I only ask you questions like that, you could if you're a good memorizer, you just memorize all the answers. And the other types of questions will be process questions. That if you didn't do it, you can't answer the questions. So if you did it in good faith, you went through it, you understood it, you took notes, you answered those questions, you should be fine. But if you didn't do it, it's to prevent someone who didn't do it from just guessing their way through. And I actually had it happen once. I used to give the Physio X exam as a separate set of exams in lab, and I had Tweedledee and Tweedledum, we'll call them, one year, somehow figured this out, and they both ended up getting 97%, and they never did one of the labs. That's what I'm trying to prevent. So, it's out there, but if you have any questions, we have the answer key. Any other questions? All right, see you on Wednesday. Yes, sir.
Is the whole test is writing, or is there any multiple choice?
There's no multiple choice. It's all big and small answer questions. Yep.
So I have a meeting.
Yep.
Is there we do the quiz another?
Yeah, whenever you want. You tell me when you want to do it.
I mean, would it be too late Wednesday?